This is arguably the most compelling evidence to date of this principle by which the brain deals with a fundamental dimension of the environment, which is time. That is that populations of neurons possess kind of rich, diverse patterns of neural activity, and how fast or slow those patterns of activity evolve provides the brain with a basis for performing time-dependent computations. In a computer, you have a central processing unit, right, which is sitting there kind of sampling a whole set of sensors or internal memory banks at some regular frequency. So there's a kind of centralized clock, if you will, right? And everything is kind of registered to that clock. In the brain, that doesn't seem to be the case. Instead, neural circuits possess this sort of rich capability of generating dynamics. So basically, if you give an input to a neural network, because neurons are connected up to each other, in many cases in a recurrent way, that creates kind of patterns of activity that reverberate in the network. It's sort of like if you were to drop a stone into a pool of water, you would see the kind of ripples on the surface. And so just by looking at those ripples and where they are, you could extract information about when, that stone had been dropped into the pool.
Basically, we train rats in these behavioral boxes that we furnish with nose ports, basically these little holes in which they can insert their snouts. And once they insert their snout on the middle one, that triggers a sound. Then there's an interval of silence, after which there's another sound. And what they then have to do is say, was the interval in between the two tones longer or shorter than 1.5 seconds? If it was shorter, they then have to move to the left, let's say. If it was longer, they have to go to the right.
Our lab was one of the first to show a close correlation between how fast or slow these ripples of brain activity evolve and judgments of time. So we looked at the particular area called the striatum that is part of a larger structure of deep nuclei within the brain called the basal ganglia. And we found that when the patterns of neural activity in the striatum evolve faster, rats are more likely to report a given time interval as being long. But when the same activity patterns evolve more slowly, they are more likely to report the same time interval as being short instead.
But correlation doesn't imply causation, right? So in order to test whether those time-varying patterns of activity were actually causing the animals, judgments about elapsed time, we wanted a way to go in and actually intervene in a very particular way, which is just to slow down or speed up how quickly the pattern of activity evolved.
And to do that, we used temperature, which had been previously used in other studies to manipulate both the temporal dynamics of behavior. So in the example of songbirds, for instance, that's in these very stereotyped songs. And by cooling a specific brain region in the bird brain, in the pathway of song production, these songs get dilated. And when you warm that region, they get contracted. But importantly, the specific sequence of syllables in the song is preserved.
Temperature gave us a knob with which we could kind of stretch or contract in time neural activity. And then when we applied that manipulation in the context of behavior, we were able to stretch and contract animals' behavioral indications of timekeeping. So their pattern of judgments about whether an interval was longer or shorter than a learned category boundary.
While rats were making these time judgments, we manipulated the temperature of the striatum. And so warming striatal tissue resulted in rats reporting time intervals as longer, and cooling did the exact opposite. They were more likely to report time intervals as shorter. And so all of these suggests that increasing temperature can be accelerating the patterns of the activity in the striatum, kind of like speeding up the dials of a clock, and so causing the rats to judge time of a given interval as being longer than it actually was.
Surprisingly, even though the striatum coordinates motor control, slowing down or speeding up its patterns of activity didn't slow down or speed up the animal's movements. So this got us thinking, maybe the striatum is important for determining what movement to do and when to do it, but not how to control those movements on a moment-by-moment basis.
So we thought maybe there's another kind of learning system for motor control that's responsible for this more continuous control and coordination. And so we've begun recently applying these same kind of temperature manipulations, now not to the basal ganglia, but to the cerebellum. And we're finding in the very early pilot data that unlike the basal ganglia, we do indeed see kind of temperature-dependent effects on the continuous execution of movement. We believe you can kind of see evidence of this division of labor between these two different brain systems, even in, for instance, the clinic. So take Parkinson's patients. Parkinson's results from largely the degeneration of dopamine input to the basal ganglia. So it's a basal ganglia dominant movement disorder. Parkinson's patients have particular difficulty in initiating movements, right? This transition from either not doing something to doing something. You might come to see the kind of constellation of symptoms that characterize diseases that clinically may appear as distinct as, obsessive compulsive disorder or Parkinson's disease is actually reflecting the same core deficit, which is a problem in being able to transition into or out of, a particular action.